Welcome to The Microscopists, a bite-sized bio podcast, hosted by Peter O'Toole, sponsored by Zeiss Microscopy. Today on The Microscopists.
Today on The Microscopists, I'm meeting with Eija Jokitalo, Principal Investigator and the Director of Electron Microscopy Unit at the University of Helsinki. And we'll hear about her passion for imaging.
I remember falling in love with microscopy in my very early studies in biochemistry.
Her outreach work helping to inspire the next generation of microscopists in schools.
I have been a couple of times teacher in this high school course, which is combining arts and science.
And we discuss her work imaging organelle partitioning during cell division.
Kind of first own projects in Helsinki were about how ER partitions during mitosis.
And the importance of alternative career paths in core facilities.
The scientific careers are very different and they need to be evaluated differently, because if you are really good in your job as a researcher in core facility, you don't have these first or last author papers.
All in this episode of The Microscopists. Hi, welcome to this episode of The Microscopists. Today I'm joined by Eija Jokitalo. I've said that wrong, haven't I, Eija?
No, it was fine.
Anyway, from the University of Helsinki, working in the Institute of Biotechnology, a good old hardcore technologist. And if you're watching on YouTube, you'll also see that Eija's got a brilliant electron microscope image in the background, which is, the more you look at it, the more you see. It's absolutely brilliant. Eija, thank you for joining me today.
Hi, nice to be here.
I think, I don't know how many years we've known each other from various meetings, but probably most noticeable from the advanced imaging courses at the VIB. I think is probably where I got to know you best from all of those, and teaching on with Chris Gary and Saskia Lippins and their teams and Sebastian Monk over there. But yeah, I'm there primarily talking about light microscopy, light microscopes, light microscopy, and you're there talking about electron microscopy in an advanced light microscopy course. So where did your passion for electron microscopy come from?
It started as a passion for imaging. So I remember falling in love with microscopy in my very early studies in biochemistry, where I did, very simple immunofluorescence imaging. And then during my PhD, I decided that I want to become an electron microscopist. So I was thinking of my kind of future postdoc position or place based on where I could kind of combine cell biology and start doing electron microscopy. And already when I left to do my postdoc, my kind of plan was to come back to Finland to start electron microscopy, kind of to land in electron microscopy. And that's what I did.
And big time. Obviously, you're very well known now in the world for your electron microscopy. I'm going to take you back though. You said that you got interested at an early age doing your biochemistry. So I presume that was in your undergraduate?
Yes.
So where was your undergraduate degree?
I have studied in University of Helsinki. So I started first year was chemistry, and then continued in the biochemistry. And so my master's is in biochemistry. And then when I started doing my thesis, there is some additional courses that you take, and then that was when I kind of specialized in cell biology. So I haven't really studied biology at all. and so I don't no names of any plants or birds or anything. So I often get a little bit kind of comments that what kind of a biologist you are. So I'm the one that kind of goes deep into the cellular level.
So how did you find that transition from chemistry to biochemistry to biology? How tough was that? Or was it easy, just a natural progression, or actually was it quite tough going?
No, for me it has been really natural, but it was also influenced by strongly by one person, Maria Makarov, my PhD supervisor. She gave some lectures about glycosylation and using yeast as a model system to study protein secretion, and I just felt that, you know, finally everything makes sense, and I went to Maria's and told her that, you know, I want to do my thesis in your lab, and she said that, yeah, I don't have any funding at the moment. And I said that, yeah, that's a pity, because I really don't want to go anywhere else. And finally she said that, okay, let's see how it goes. And the first year I had, like, you know, grants for always couple of months. Yeah. But by the end of the first year, Maria got a kind of a big research grant, and actually then could kind of give me a contract to the end of the PhD project. So I was in the end, you know, lucky. But it was kind of the idea that at that time it made so much sense to use yeast as a model for cell biology. It was so easy to, you know, delete genes and study the effects. And I think that that was the time when I was kind of guided to secretory pathway, and I'm still there. So, you know, in the picture you see two of my favorite organelles, ER and Golgi. And I started studying protein folding in ER during my PhD, and then I went to do my postdoc in Graham Warren's lab in London. And there I studied the partitioning of Golgi during mitosis. And then instead of going further on the secretory pathway, I actually ended up in the retrograde transport route and was transported back to the ER. And in Helsinki, when I started my own research team, I kind of continued a little bit in the same theme as in Graham's lab, so studying organelle partitioning during mitosis, but going back to the ER. So my kind of first own projects in Helsinki were about how ER partitions during mitosis. And since that, it has been kind of first quite concentrated on ER, and lately now more on in general, kind of how the organelle shape supports its functions.
So I've got two questions on the back of that. So I'm going to ask the scientific one first, actually. So what's your bigger passion? The advancement and utilization of electron microscopes, or that process going to ER and mitosis?
They are totally connected.
Oh, very carefully.
I'm kind of my own, the most kind of the biggest research topic I can kind of think of is the organelle shape and structure. But I wouldn't like to study that using any other tools. So imaging and visualization, seeing them is the key. So as a microscopist and I'm leading a EM core facility. So we are my facility, we have around 100 projects each year, and we also do a lot of collaboration projects. So through these collaborations, I'm studying kind of really wide range of model organelles, organisms, different kind of research questions. And this is really fascinating too, but always when there is a chance of making a detour to ER or Golgi, I will take it.
So I thought the one bit that was interesting there, well, it was all interesting. The one bit I think to pick up on those, you have 100 projects going through using the microscopes. It's a huge number of projects, and the electron microscopes, the importance of different technologies underpinning science. And I would imagine the range of scientific questions you're addressing using the electron microscope in those 100 projects will range from cancer research to neurobiology to the fundamentals of developmental biology, microbiology. You'll probably encompass all those aspects. So yeah, I guess some people go into science wanting to do cancer research because they're very passionate for personal reasons about cancer or Alzheimer's or whichever else. And you're in a position, as with many core facilities across the world, to actually be helping all of those type of studies. And without that expertise in electron microscopy, it couldn't be done. How hard do you find it balancing all those projects and allowing them time, equitable time on the microscopes, staff support time, because some of these applications are quite difficult and they need expert hands, and that all needs funding as well as a direct grant. How do you find balancing all of that?
So in my core team, I have 3 technicians and 2 researchers, and out of these 100 projects, bit over half of them is just providing service. So my technicians are providing service on sample preparation, making thin sections, and then we provide training for the users to use the microscopes. And actually, the electron microscopes are much way easier to use than light microscopes. So it doesn't take that. We spend roughly 5 hours per person providing the training, and in that time they learn how to put the sample in, focus, select the magnification, take photos, which is kind of the basic things they need to learn to be able to take the pictures. And then again, they have to spend time in learning how to interpret the data. And these again we provide in kind of support. But in these kind of service projects, we are not scientifically involved, so we just provide the support. And then on the collaboration projects, we are scientifically involved. And there, the key is kind of communication. We are good what we do. We know how to interpret the data and take the pictures and recommend what kind of sample prep to use. But at the same time, we don't know the research questions, and we cannot be experts on every model organism there are. So the successful projects are where we really communicate well, and the collaborator understands they model and they research questions, and then we bring the technology, and then when we kind of operate together we actually get the results.
And so you have 3 technicians, 2 research officers within it.
Yes.
And these career paths are, in the grand scheme of time, a relatively new type of positions within universities. And it's interesting to get an understanding of the type of person that wants that type of role.
Before going to that, you know, the success, why I'm kind of really happy at what I'm doing is that actually, you know, I have a great team. So 2 of my technicians have been EM technicians over 30 years. And the third technician actually is my former postdoc. So she has also a long kind of experience in and then my researchers, Ilya and Helena, they both also have been in the unit over 10 years. So we have really developed things together. And it's very different from the time when I started. But you are correct, the infra personnel is kind of a new concept. And in Helsinki University, I have been leading a working group who have been making a kind of career model for infra personnel. And it was beginning of February this year, we actually presented it to our institute board. And now the university HR is kind of working based on this model. And some of the key issues there are that we actually in core facilities, especially the scientific careers are very different, and they need to be evaluated differently, because if you are really good in your job as a researcher in core facility, you don't have these first or last author papers, but you have actually a lot of these papers where you are in the middle. And actually then the way to evaluate how successful you are is asking the questions that what was your impact on this paper, that without your work and without this EM part, you know, would this paper have been accepted at all, or you know, how much it raised the level of the paper. So we have to even kind of formulate different kind of criteria how to evaluate the persons, you know, when thinking how they kind of progress in their career. And of course, this has been a little bit the same for me. I'm kind of evaluated as through my scientific achievements, and then sometimes it's you know, not so clear that how much credit I can take from all these collaboration projects, or should is my scientific profile just, you know, the work that we do in my tiny group.
I think I'm glad you mentioned this. I'll bring something up in a moment that I'm doing at work at the moment. I think the scientific profile is not just on your own science, it is on the facility, it's on your impacts and expertise in the microscopy, which is academic in a way as well. And so I'd say they go hand in hand. I'm just looking at job descriptions at the moment and for new staff coming in, and there's templates for technical staff, there's templates for postdoctoral staff, but actually a lot of our staff are technical staff, but they have that academic trait to them, and they have to have that academic side to them as well. So they kind of sit in between these two groups. Now, I would say they are more on the technical side, and I think we should say that with pride. I think that's a really good role, because we are multitaskers. But at the same time, many of our staff are postdocs to hire. You know, they become a group's postdoc for that moment in time where they need that expertise.
Yes.
And how you evaluate that, even how you put a job description for pay scales, is very difficult.
Yes, this is we need to kind of create this third personnel category, and this is actually what we proposed very clearly, that we kind of made a list of job titles, but we also made a very kind of bold proposal that we really need to have, you know, a third personnel category created, and that of course, you know, involves a lot of legal technical things. It will take several years, but it's kind of how in the future, you know, things should go. In the core facilities, you know, we provide service, we maintain instruments, very expensive instruments, but we also do research and we develop things. So there is both the technical aspect and then the research aspect.
I'd be interested to see that document at some point. It's something I'm just playing with at York at the moment, and York has fantastic scales. They're very on par, and it's well rewarded. But I do think there's an opportunity to look at if we can make it more clear, operational scales and so forth as well. Going back, you said you did your PhD in London.
Postdoc.
Sorry, postdoc in London.
Yes.
How did you find living in London compared to Helsinki?
Very, very different. But yeah, I actually, when I came to London, I came there with my 7-year-old daughter as a single mother, and I had an au pair, and you know, it wasn't that easy, but the work itself was so interesting that I still think that those 3 years were kind of the, you know, the peak of my my time. I really we found in London a kind of corner in the kind of northeast where there was kind of the Epping forest started. So it was a little bit like, you know, similar what we have here. So every Sunday we could kind of go for walks on a forest, and it wasn't that crowded and kind of.
You found it. So I was going. One of the questions I was going to ask you, when was probably the most fun time of your career? But it sounds like that was probably your.
Yes, there was so much kind of. You know, we were a big lab. We were 15 postdocs, and everyone was fully committed to work, and we really worked hard. But also the you know, we had a freedom to start the projects and see where they kind of take us, and of course the resources were, you know, we had our own transmission electron microscope just, you know, for the lab. So I was sharing that microscope with one technician and one student. So no heavy booking.
Yeah, but now how many electron microscopes do you have?
Okay, we have 5.
Yeah, see, even better. Just lots of users.
Yeah, but not, you know, there's much more users. And of course nowadays, you know, I don't get to do, you know, the things I'm best. So I'm my you know, nowadays I'm just kind of following from the back when my students or you know, someone else is doing the fun things.
So how did you find that transition of moving from the driver to the backseat driver to the probably the person back in the taxi rank just telling the drivers what they should be doing and hearing where they need to go next?
Well, it happens gradually, so you know, you don't notice it in a way. it's kind of, I think it's quite natural development.
And do you enjoy the way it's evolved?
Yes, because there has been other things that has come along. Of course, yes, it's stressful to make applications and reports all the time, but for example, I think that one of the really best features in core facilities is kind of the networking. So in Finland, we are a small country and we don't have that much resources. So I think that quite early on, 2009, we formed these kind of Biocenter Finland networks. And for example, in that, in all of the universities in Finland, the imaging units were kind of networked. And since that time, we have jointly made our strategy that who specializes in what and what kind of instruments we apply. And for example, in that strategy, we decided that we want to specialize on 3D EM, and then the others have supported us in the applications to get the microscopes for that. And then at the same time, I know that I don't need to provide everything for our users, that there are some techniques where I can then just send our users that, why don't you contact? And this way, we are not kind of competing with each other. We are competing against the other networks and platforms, but we have this strategy, and it's something that has come to replace kind of that. Yes, I don't have time to make experiments in the lab anymore, but it's kind of a new type of thing which I also enjoy a lot.
So have the mass spec community, the genomics community, have they got their acts together in such a good way as well, or are they still very much?
Yes, we in this Biocenter Finland network, we have 19 platforms. So basically,
because that means you're competing against them and they've got their act together as well. Otherwise you
yes. But so the in the you always have that network support, and everyone's doing this kind of strategic. So it's easier to compete as one out of 19 than alone with these 58 applications. And then it's much more like on the hands of the evaluators.
So thinking of networks, it's not just the national network, international networks are equally important. So what's your favorite meeting?
I have this dual thingy in everything. So as a cell biologist, it's the ASCP, which I try. I don't participate every year, but I try to go as often. So it's great. I like the kind of the atmosphere and seeing people and also meeting all kind of ex-postdoc friends and So and then as a kind of microscopist, so the Scandin meeting.
Yeah.
That's for a social eventing.
Yeah,
so
that's always based around Scandinavian groups.
Yes. So that's just meeting all the vendors that represent kind of Nordic countries, and then meeting all the colleagues So that is mostly like, you know, the events that kind of happen outside of the lecture rooms, and then the every second, or in a way, every fourth year is the International Electron Microscopy Society meetings, and then every fourth year is the European counterpart for it. So these are big meetings which have kind of really big instrument exhibitions, and then everyone tries to come. And of course, you know, the international meetings have provided me a way to travel to, you know, everywhere in the world. I have been in, the first one I participated was in South Africa, in Durban, 2003. And then since then I have been in Brazil, in Rio de Janeiro, in Japan, in Sapporo, in Sydney. Last time it was in Sydney, and then in Prague.
So where was this one?
That's in the Sapporo meeting. So that's a lunchbox, and we were starting at this kind of lecture seminar, and I was kind of very, very proud of being able to use the chopsticks with some noodles.
And on your lap as well, which is cool. So IMC and EMC, and of course, I guess I'm going to tell you off now. The first thing you should have said was EMC is my favorite one, obviously, and come over, because you're helping run one, aren't you? Coming up soon.
Yes, so I'm in the organizing team for the next EMC meeting. It was scheduled to happen in August 2020, and it was unfortunately cancelled quite in the last minute. We had already kind of abstract submissions going on, and so on, and it was delayed for 4 years. So hopefully the next meeting will be in 2024 in Copenhagen. So Scandem, Scandinavian Microscopy Society, is kind of the host organizer, and I'm chairing the life science symposium. Yeah. So it's a huge privilege, but it's also, again, a great way of networking and really connecting with people all over Europe and making plans.
Yeah, so I very much looking forward to it, because I was going to 2020 and I look forward, hopefully we'll get to 2024 for it. I know obviously Klaus, Sandra, Julia are all part of that team that are helping assemble it as well. Now, you sent me another picture, which is which looks like a map of the world, but I'm not sure it is. What is it?
Yes, it is. It is a picture made by a high school student, Evi, and this picture is one. It's actually a framed picture on the wall of our unit, and another kind of new or different kind of feature in my work is that I host visits for the high school students. So when they just come to visit the university, and you know, when they are kind of preparing themselves to the entrance exams for the university, and it's always kind of fun to see these students who get younger every year. But in addition, I have been. It started after one of these visits, and then developed into actually a course. So I have been a couple of times teacher in this high school course, which is combining arts and science. So it's actually, it's me and the school's art teacher, biology teacher, and physics teacher. And the physics part, of course, is kind of thinking how the microscopes operate and kind of going a little bit into physics behind the imaging. And then the biology is actually the teacher and the students, they bring different types of samples, which one afternoon in my lab we prepare. Typically we prepare SEM samples, and then we select from our collections some sections, and then another afternoon the students can use all of our microscopes. So they can take whatever kind of images they want, and then the third part is making art of it. So we provide little bit introduction to these imaging software kind of, and in this picture Evi has it is a kind of thin section image of a cultured cell, and then she has colored it to look like a map.
So you are letting high school children use your microscopes, which cost how much money?
A lot. the one we got last summer was 960,000 euros. So close to €1,000,000. The TMs, the standard TMs are not so expensive. You can get a really nice one and a camera with 500,000. But of course, we have someone there to watch.
Yeah,
so we don't just, yes. So the students, actually one challenge with the students is to make them to remember that they will use the microscope camera. Because what is quite often happens is that they have their cell phones and they take pictures from the monitors. And I try to tell them that, I'm providing you access to this 200,000 euro camera. Why don't you use it instead?
Isn't that great though? Because it shows they're engaged and they want a picture to take home to show their parents or their friends. Say, look what I've seen, look what it is. Because come on, we all take pictures of everything these days, don't we? And go, look what I saw. They'll put it up on social media, and I think that's great. And I love the contrast to a £200,000 camera on your TM to your a few euros worth of camera that's on those. But you're also into photography as a hobby.
Yes, but I'm and that's one picture. It's kind of a reflection of some moss growing on a tree on the river, and what I like this is that it's not so easy to say that what is kind of the real thing and what is actually the reflection of the water. So I'm not good in photography, that's for sure, but this is one of the, if I look the pictures I have taken, I think that almost half of them are either running water or some kind of reflections on the water. I don't know what fascinates me on those.
So is this yours as well?
Yes, this picture I chose to, that's where I live. So I live in this really far east corner of Europe where sea freezes every winter. And this is actually a sunset on a frozen sea. And the picture is taken around 2:30 in the afternoon. So in December, the sun goes down before 3 p.m. And I don't think, well, we can see on the background, there is already some snow.
Yeah.
The sea is maybe not so thick, the ice yet, that I didn't want to walk on it, but around January time you can drive your car on the sea.
So how have you found, and you obviously got a daughter, how old is your daughter now?
Sorry?
How old is your daughter now?
My daughter is 33.
33, and just the one daughter?
Yes.
Okay. How did you find in the early parts of your career balancing your work life? Bringing up, you said you're a single parent, bringing up a child, you're doing your postdoctoral research, developing your academic career on the back of that, after that. How did you find balancing all that?
Well, you know, again, it was very natural because she was born, you know, before I kind of became a researcher. She was born already during my master's studies. So it has been all the time, you know, the two of us. And
what has she got on to do?
Well, she's a vet.
Ooh. So keeping some sort of science in the family.
Well, she's doing. She works in a hospital, so yeah, she not a scientist, but really doing clinical work. But she's actually in UK now. She moved in the beginning of the year to do a specialization on ophthalmology.
Okay.
In UK. So
whereabouts is she?
In Portsmouth.
Okay.
Or haven't. So very south.
Yeah.
So, yeah.
I have a niece who's a vet down the south, but not in Portsmouth.
Yeah.
So,
yeah.
But that's really cool. You must be very proud of her as well, and succeeding through. So your other hobby is patchwork.
Yes, I'm doing any type of handicraft, but that's like art quilt. Yes, so some embroidery and things. Anything to do with manipulating fabrics. I love colors and then sewing and just doing things. And I think that's kind of the balance to the scientific part.
How did you take the picture? Because I can see one hand, I can see another finger which is on the other hand.
That's not. It's someone. My friend has taken the picture, so it's my fingers and yeah.
I was just trying to work out. So one hand, I can see another finger, so two hands and a picture. I thought that's incredible.
Yeah. I belong to this group of 10 women, and we meet. We are kind of around Finland. We meet a couple of times per year, and we organize one kind of exhibition roughly per year. So during these meetings, we plan the theme and do kind of practical plans, and then everyone is doing the kind of art pieces solo. And then we meet again when we kind of set up the meeting, and that part is actually we spend 4 days in a small island, and of course when going to an island you cannot take a sewing machine or much of the material with you. So the instructions were that everyone takes a white shirt and then a couple of needles and then some thread. And then over these 4 days, we were just going around the island, enjoying the views and staying wherever and sewing. And then, you know, in the end, these shirts were not really looking shirts anymore.
a truly well knitted network, one could say. Eija, I'm going to ask you some quick fire questions. So, PC or Mac?
PC.
I knew it'd be that. McDonald's or Burger King?
McDonald's. I don't like smoke at all, so Burger King. I don't. Yeah, it's smoked stuff. No.
That's fine. Early bird or night owl?
Night owl.
Oh, okay. I'd say just like Lucy Collington. So, okay, still a good electron microscopist in the dark, I'm sure. Tidy or messy as a person?
Tidy.
Tidy. Maximist or minimalist?
Maximist. Everything's kind of full of stuff. Yeah.
Tidy maximist. Impressive. Okay, tea or coffee?
As you don't know, the dirt looks really ugly in microscope. So we need to keep things tidy.
No, that is very true. Tea or coffee?
Tea, never coffee.
Oh, chocolate or cheese?
Chocolate, never cheese.
I like this certainty. This is not.
Yes.
TV or book?
Audiobook.
Audio. Good grief. So get in the middle. Audiobook. What's your favorite film?
Baghdad Cafe.
Sorry?
Baghdad Cafe.
I don't know it. Oh no. Okay. That's one to look up. What's your favorite Christmas film?
I don't have any.
Oh, okay. What's your pet hate?
What my pet hates?
Yeah, what annoys you?
When they vomit. I don't like cleaning after them.
That's fine. What do you most love? What's an item that you would, a luxury item that you most love?
My sewing machine.
Oh, good
I
answer.
have a really fancy one.
Good answer. So next question, what is your favorite item of clothing then?
It must be some of my woolen socks.
Oh.
I have like a huge basket of them. So
Of woolen socks?
Yeah.
All paired up together?
Yeah.
Nice. Do you prefer to cook or to be cooked for?
I like to control things, so I would prefer cooking.
Okay, and what is your signature dish then? What is your best dish that you cook?
Anything, depends who's coming.
Okay.
For my sister's kids, they want to have, they prefer meatballs. My daughter is a vegan, so some vegetarian. If I ask my lab to visit, it would be some kind of a meat thingy in the oven.
Okay. What is your least favorite food?
Well, I'm allergic to fish and seafood, so anything, and also seaweed. So basically anything coming from sea.
Okay. Ooh, that's not so easy. Did I ask you? I don't think I asked you. Wine or beer?
I think beer goes into the same category as coffee. So it's just the bitterness that feels strange. There's actually, I'm collecting statistics. So usually people who don't drink coffee, they don't like beer, and vice versa. So it's not easy to find a coffee lover who doesn't drink beer.
So it's wine, red or white wine?
Only white.
Only white?
Yes.
Because the red's too bitter?
Yeah. My taste buds are like, from the 10 years old, I guess. Actually, you started with thinking what was when we met properly the first time, and from this Ghent meeting, what I remember best is us sitting in this really nice restaurant with Chris and Tom and listening to they stories about great wines.
Yeah.
And I had the feeling that we were kind of the listeners in these stories. So Chris and Tom were kind of the really wine.
Yeah, so that was Chris Guerin and Tom Derring from
Yes.
Mark Ellisman's lab, wasn't
Yes.
it? And
Yes.
but they like their Californian wines, and they know their.
Yes,
yeah,
but it was great stories. It's you know, interesting to listen to people who are enthusiastic over, you know.
But tell me, were you not sitting there drinking your wine, listening to them talk about it and thinking, well, talk about it, but can you order the next bottle, please? We were not thinking that. Oh, Chris be mortified because Chris never lets it go empty. Oh, I'm joking, obviously. Okay, so moving forward, I think your inspiration was probably one of your early supervisors from what you said earlier. What would you say your favorite publication is that you've authored or co-authored for whatever reason? Do you have a favorite publication?
Yes, I think it is the first one where I'm the last author. My first student, Maija's first paper, which was published in JCP in 2007, where we showed how ER partitions, or that ER undergoes this kind of structural transformation.
That's a very nice answer. It's good, that first out, your lab, your group, your research. That's really cool.
I haven't supervised that many students kind of on my. I have co-supervised a lot of my kind of students also from my collaborators, but I have 4 students, you know, fully kind of responsible, and I'm so proud of each of them. You know, I think those are kind of the really great moments, and I think those are the biggest achievements in a way of really supervising, you know, the kind of making new researchers and guiding them to this field.
Yeah, which is a good point. That's an important part. You've got an academic career, which is doing your primary research and developing it, but at the same time, you're developing careers alongside that. And as you've already mentioned, you do your outreach alongside that. And I think this is a picture of your group.
Yes. This is in the middle. There is these 2 guys, Behnam and Darshan. They are my students. And then the rest is my EM facility team, and we are all in the same boat. Unfortunately, the boat was so small that it was safer to stay, you know, on a crowd. We didn't go to the lake, but
so quite literally. So for those who are listening, they are all in the same very small boat, which is quite a task for eight people. At the same time?
Yes.
But you're not in it.
I am there in the middle with the black hair or dark hair.
You can't change the color of your hair. That doesn't help.
I'm changing it all the time.
So who took the picture then?
This picture was taken in Jyväskylä during the dinner in Scandem meeting. So
there's plenty of...
Yeah.
Just in case they sailed off with it, you need to be careful on that. Where do you see the next big challenge for microscopy? What needs to change? What would be the next big step if you could?
This is a very personal view, and not really. I'm sure it's not the right answer, but what I see now, that there is a trend that we try to automate everything, and we go bigger and bigger. We want to collect always bigger volumes and higher resolution. And I don't think that that increases the understanding of the scientific question necessarily. So I don't kind of, I don't like that trend so much. So I for example, I'm waiting that someone kind of reinvents stereology again, like 800, kind of mathematical approach which has been around long time. So instead of everyone trying to just make large 3D volumes and segment objects, I think that we could actually use stereology and start analyzing, and get. I love pictures. I want to see everything, and in the end, I want to make them into numbers. And I think that we should remember that in EM, the power is that we see the structures and in the right surroundings, and we should pay attention to analyzing the structures and their surroundings, and try to go kind of, not to decorate things, but really measure and analyze things. And
they can go hand in hand. Do you not think that by getting more volumes, bigger data sets, you can make more measurements in a more quantitative way?
Yes, but is that always necessary? In the stereology, it shows that you can actually, instead of calculating areas from every slice, you take every 20th slice, and you actually can quite nicely predict. So the same is little bit that, I think that I think all these correlative things is the key. So we should always kind of get the bigger picture with something else, and then EM for the details. And yeah, so maybe that would be maybe the right answer from me, that where we're going is to correlative imaging. So one imaging technique is not going to solve everything, and there isn't one over the others. But really what we should kind of, and what we have already, of course, the work has started. So we're trying to connect and combine the different imaging modalities.
Yeah, so going from the correlative light to electron microscopy, which of course then gives you the volume, the size, the proportion. You left maybe the electron microscope to concentrate, focus to where it's really.
And we have also tried to connect MRI imaging and this diffuser tensor imaging with 3D EM, where we get kind of functional data from the MRI images from some brain activities, and then go from there to EM.
Which is, for anyone listening who doesn't understand the technologies around that, linking from an MRI to a light microscope is very challenging. To go from an MRI to an electron microscope, which almost bypasses that middle bit, is incredibly challenging. And Eija, we are now up to the hour mark. Oh yeah, that has gone so stupidly fast. I've just got to ask you two really quick questions. What did you want to be when you were 10? Around that, as a young girl?
Hairdresser.
Hairdresser.
Mm.
Good answer. What would you be if you could be anything today?
I think I have pretty much, I'm doing what I wanted to do. I have followed one path. I haven't just drifted to what I'm doing, but I really, I don't think I have other options.
Forget options.
When I started studying, I was thinking that would it be some kind of crafts or science, and it was a great advice from my mom that you also need a hobby, that there is a risk if you making, you know, your most passionate hobby as a kind of profession. How do you then balance, you know, things?
I love that answer, because that's why I never became a professional footballer, simply because I thought I should balance
Yes.
my career.
Yes.
There might have been other reasons as well, but I'll just go with that one. Eija, on that note, thank you very much for joining me for this version of the podcast. Everyone who's watched or listened, please don't forget to subscribe to whichever channel you're listening to. And actually, you've heard Eija talk about all sorts that Lucy Collins has talked about. Jeff Lippincott-Schwartz is part of the ASCB and helps lead all of that. Jeff Lickman does that big volume EM, so there's plenty of other podcasts you can go back onto to hear similar content. But you know, I really enjoyed hearing about the career progression, the importance of jobs within this type of area of research and facilities, and the challenges that you are trying to solve in Helsinki itself. And actually not just saying there's a problem, but actually doing something about it, and then taking it up to senior management, say, look, here we are, this is what we need. And the fact that they're empowering you to do that, I think, is a great credit to Helsinki as well in supporting that. So, Eija, I can't wait to catch up soon. Thank you.
Okay, see you soon. Bye.
Thank you for listening to The Microscopists, a bite-sized bio podcast sponsored by Zeiss Microscopy. To view all audio and video recordings from this series, please visit bitesizebio.com/the-microscopists.